Endoplasmic reticulum. Endoplasmic reticulum, it is a network of interconnecting membranes. It is continuous from outer nuclear envelope to outer plasma membrane. It is reticular arrangement. It is prominent in cells which are actively synthesizing proteins. Proteins, glycoproteins, and lipoproteins are synthesized in the ER. Proteins, glycoproteins, and lipoproteins are synthesized in the endoplasmic reticulum. Detoxification of various drugs is an important function of ER. Detoxification of various drugs is an important function of endoplasmic reticulum. Endoplasmic reticulum is classified into two varieties: rough endoplasmic reticulum and smooth endoplasmic reticulum. Endoplasmic reticulum looks rough due to ribosomes. It is related with protein synthesis. Endoplasmic reticulum looks rough due to ribosomes. It is related with protein synthesis. Smooth endoplasmic reticulum is related with lipid synthesis. Mitochondria. Mitochondria is spherical, oval, or rod-like bodies. Mitochondria have two membranes, inner membrane and outer membrane. The inner membrane having folds also known as cristae. The inner membrane having folds also known as cristae. The inner membrane contains the enzymes of electron transport chain. The fluid contains the enzymes of citric acid cycle and urea cycle, etc. Mitochondria are the powerhouse of the cell. Mitochondria are the powerhouse of the cell. In mitochondria, different metabolites are oxidized and energy is stored in the form of ATP. Erythrocytes do not contain mitochondria. Erythrocytes do not contain mitochondria. Cytochrome P450 system present in mitochondrial inner membrane is involved in steroid synthesis. Cytochrome P450 system present in mitochondrial inner membrane is involved in steroid synthesis. Mitochondria also contain specific DNA. Mitochondria also contain specific DNA. Inner membranes protein are made by own DNA. Inner membrane proteins are made by own DNA. Other majority of protein, especially of outer membrane, are synthesized from cellular DNA. Plasma membrane. The plasma membrane separates the cell from the external environment. It is semi-permeable, so entry and exit of compounds are regulated. The membrane is metabolically very active. Structure. Structure of the membrane was described as a fluid mosaic model by Singer and Nicholson. Membranes are mainly made up of lipids, proteins, and small amount of carbohydrate. Membranes are mainly made up of lipids, proteins, and small amount of carbohydrate. The contents of these compounds vary according to the nature of membrane. Membrane carbohydrates. Carbohydrates are present as glycoproteins and glycolipids. Membrane lipid. Phospholipids are the most common lipids. The phospholipids are arranged in bilayer, polar head arranged towards outside and hydrophobic parts towards inside. Polar head arranged towards outside and hydrophobic parts towards inside. Cell membranes also contain cholesterol. The lipid bilayer shows free movement of its components, so it is also called as fluid in nature. The lipid bilayer shows free movement of its components, so it is also called as fluid in nature. Membrane proteins. The peripheral proteins are situated on the surfaces of the bilayer. The integral membrane proteins are situated inside the bilayer. The integral membrane proteins, which are arranged in whole bilayer, is called transmembrane proteins. The transmembrane proteins can work as receptors. The transmembrane proteins can work as receptors. Classification of amino acids. Classification of amino acid based on structure. Number 1, A. Aliphatic amino acid. Monoamino monocarboxylic acid. In aliphatic amino acid, monoamino monocarboxylic acid. In that simple amino acid, glycine, alanine, simple amino acids in them, glycine, alanine, branched chain amino acid, valine, leucine, isoleucine, branched chain amino acids, valine, leucine, isoleucine. Hydroxy amino acid, serine, theronine, hydroxy amino acid, serine, theronine. Sulfur containing amino acid, cysteine, methionine. Sulfur containing amino acid, cysteine, methionine. Amino acids with amide group, asparagine, glutamine. So, in monoamino monocarboxylic acid, simple amino acid glycinaldine, branched chain amino acid valine, leucine, isoleucine, hydroxy amino acid serine, theranine, sulfur containing amino acid cysteine and methionine, amino acids with amide group asparagine and glutamine. Monoamino dicarboxylic acids in aliphatic amino acid, monoamino dicarboxylic acid, aspartic acid and glutamic acid. Dibasic monocarboxylic acid, lysine, arginine. Dibasic monocarboxylic acid, lysine and arginine. So, aromatic amino acid, phenylalanine, tyrosine. Aromatic amino acid, phenylalanine, tyrosine. Heterocyclic amino acid, tryptophan, histidine. Heterocyclic amino acid, tryptophan, histidine. That amino acid, proline. Amino acid, proline. Derived amino acids. So, in derived amino acid found in proteins, after the synthesis of proteins, some of the amino acids are modified. Hydroxyproline, hydroxylysine are important components of collagen. Gamma carboxylation of glutamic acid residues of proteins is important for clotting process. Gamma carboxylation of glutamic acid residues of proteins is important for clotting process. In ribosomal proteins and in histones, amino acids are extensively methylated and acetylated. Derived amino acid not seen in proteins. Non-protein amino acid, some derived amino acids are seen in free in cells, that is ornithine, citrulline, homocysteine. These are produced during the metabolism of amino acid. Thyroxine may be considered as derived from tyrosine. Non-alpha amino acid, gamma amino butyric acid is derived from glutamic acid. Non-alpha amino acid, gamma amino butyric acid derived from glutamic acid. Beta alanine, where amino group is in beta position, is a constituent of pantothenic acid, vitamin coenzyme A. So, gamma amino butyric acid derived from glutamic acid, beta alanine, where amino group is beta in position, is a constituent of pantothenic acid and coenzyme A. So, this is one in one aliphatic amino acids we discussed. Aliphatic amino acids include monoamino monocarboxylic acid, monoamino dicarboxylic acid, then aromatic amino acids, phenylalanine, tyrosine, heterocyclic amino acid, amino acid, derived amino acid found in protein, derived amino acids not seen in proteins. So, non-alpha amino acids. Even what do you say? Aliphatic amino acids. So, number one, A, aliphatic amino acid, B, aromatic amino acid, aliphatic amino acid, aromatic amino acid. In aliphatic amino acid, monoamino monocarboxylic acid, monoamino dicarboxylic acid. Then aromatic heterocyclic amino acid, amino acid derived from derived amino acids, derived amino acid found in protein, derived amino acid not seen in proteins. Then non-alpha amino acid. Then classification based on side chain. Classification based on side chain. Amino acids having non-polar side chains. Amino acid having uncharged or non-ionic polar side chains. Amino acids having charged or ionic polar side chains, hydrophilic. So next, first, amino acid having non-polar side chains. These include alanine, valine, leucine, isoleucine. Isoleucine. Aromatic amino acid, methionine, proline, phenylalanine, and tryptophan. These groups are hydrophobic, water repellent, and lipophilic. Therefore, the parts of proteins made of these amino acid will be hydrophobic in nature. Amino acids having uncharged or non-ionic polar side chains. Glycine, serine, threonine, cysteine, tyrosine, glutamine, and asparagine belongs to this group. Glycine, serine, threonine, cysteine, tyrosine, glutamine, and asparagine belong to this group. These amino acids are hydrophilic in nature. Tyrosine and cysteine may show hydrophobic character when present in the interior of the protein. Amino acids having charged or ionic polar side chains, hydrophilic. So acidic amino acid, they have negative charge on the R group, aspartic acid and glutamic acid. Tyrosine is mildly acidic. Basic amino acid, they have a positive charge on the R group, lysine, arginine, and histidine. Classification based on metabolism. Purely ketogenic. Leucine is purely ketogenic because it is converted into ketone bodies. Ketogenic and glucogenic. Lysine, isoleucine, phenylalanine, tyrosine, tryptophan are partially ketogenic, partially glucogenic. However, in human, lysine is predominantly ketogenic. Human lysine is predominantly ketogenic. During metabolism, the part of the carbon skeleton of these amino acid will enter the ketogenic pathway and other part to glucogenic pathway. Purely glucogenic. All the remaining 14 amino acids are purely glucogenic as they enter only into the glucogenic pathway. Classification based on nutritional requirement, essential or indispensable. The amino acid may further be classified according to their essentiality for the growth. That's isoleucine, leucine, threonine, lysine, methionine, phenylalanine, tryptophan, and valine are essential amino acids. Their carbon skeleton cannot be synthesized by human beings, and so preformed amino acids are to be taken in food for normal growth. Partially essential or semi-essential. Histidine and arginine are semi-indispensable amino acid. Growing children require them in food, but they are not essential for the adult individual. Non-essential or dispensable. The remaining 10 amino acids are non-essential because their carbon skeleton can be synthesized. So that's all about amino acids. Then the structure of proteins, organization of proteins. Proteins have different levels of structural organization. Primary structure of protein means order of amino acids in the polypeptide chain. Secondary structure means the relationship of amino acid close to each other. Tertiary structure means interrelationship of various regions of a single polypeptide chain. Quaternary structure results when the protein is made up of more than one polypeptide chain joined together. Number one, primary structure. Primary structure of protein means the order of amino acids in the polypeptide chain. Each polypeptide chain has a unique amino acid sequence. That sequence is decided by the genes. The primary structure is maintained by covalent peptide bonds. The peptide bond is a partial double bond. Numbering of amino acids in proteins. In a polypeptide chain, at one end there will be one free amino group. The end is called amino terminal or N terminal, and the amino acid is named as first amino acid. At the other end, the chain is there is a free carboxyl group. It is known as carboxyl terminal, that and the amino acid is named as the last amino acid. Other amino and carboxyl groups are involved in the peptide bond formation. Primary structure determines the biological activity of protein. Higher levels of structural organization are dependent on the primary structure. Even a single amino acid change can affect the function of protein. For example, HbA, normal hemoglobin, the sixth amino acid in the beta chain is glutamic acid changed to valine in HbS, sickle cell anemic people, normal hemoglobin HbA gene and HbS gene in sickle cell anemic people. Secondary structure of protein. Secondary structure means the relationship of amino acid close to each other. Secondary level of protein structure is maintained by non-covalent forces described below: hydrogen bond, ionic bond, hydrophobic bond, van der Waals forces. Alpha helix. The alpha helix is the most common and stable conformation for a polypeptide chain. The alpha helix is a spiral structure. The structure is stabilized by hydrogen bonds between the NH and CO groups, carboxyl group of the amino and carboxyl group of the amino acid. Each turn is formed by 3.6 residues and each turn is 5.4 angstrom. The distance between each amino acid residue is 1.5 angstrom. The alpha helix is generally right-handed. Proline and hydroxyproline will not allow the formation of alpha helix. Beta pleated sheet. In beta sheet, the distance between the adjacent amino acid is 3.5 angstrom. It is stabilized by hydrogen bonds between NH and carboxyl group of the neighboring polygenic polypeptide segments. Parallel beta sheet. If adjacent strands in a sheet run in the same direction with regard to the amino and carboxy terminal ends of the polypeptide chain known as parallel. Anti-parallel beta sheet. If adjacent strands in a sheet can run in the opposite direction, known as anti-parallel beta sheet. Beta pleated sheet is a major structural organization in proteins like silk fibroin. Beta sheets may be formed in many proteins by the U-turn folding of the chain. Tertiary structure. Tertiary structure means interrelationship of various organs of a single polypeptide chain. The tertiary structure denotes the three-dimensional structure of the whole protein. The tertiary structure is maintained by non-covalent interactions such as hydrophobic ends, electrostatic bands, and van der Waals forces. Domains are generated in tertiary structure. Quaternary structure. Quaternary structure results when the protein is made up of more than one polypeptide chain joined together. The quaternary structure is maintained by hydrogen bonds, electrostatic bonds, hydrophobic bonds, and van der Waals forces. Depending on the number of polypeptide chain, the protein may be termed as monomer, 1 chain, dimer, 2 chain, tetramer, 4 chain. For example, 2 alpha chains and 2 beta chains form the hemoglobin molecule. Next, denaturation of proteins. Change in protein structure is known as denaturation. It occurs due to mild heating, treating with acid and alkali, X-ray and ultraviolet rays. There will be alterations in secondary, tertiary and quaternary structure of protein molecule. Primary structure is not changed during denaturation. Primary structure is not changed during denaturation. Due to denaturation, solubility is decreased. Precipitability is of the protein is increased. Biological activity of protein is lost. Denatured proteins have more sites for enzyme action, so easily digested. Denatured proteins have more sites for enzyme action, so easily digested. Cooking leads to denaturation of proteins, so cooked foods are easily digested. Cooking leads to denaturation of proteins, so cooked foods are easily digested. Most of the time, it is irreversible process. Denatured proteins are sometimes renatured when the physical agent is removed. There, for example, hemoglobin denaturation can be reversed. Classification of proteins. The following classification are given: classification based on functions, catalytic proteins, which they are enzymes, structural proteins, they are collagen, contractile protein, myosin and actin, transport protein, hemoglobin, myoglobin, albumin, regulatory proteins or hormones, ACTH, insulin, growth hormone, genetic protein, histone of DNA, protective proteins, immunoglobulins, antibodies, interferons. So, catalytic protein, structural protein, contractile protein, transport protein, regulatory protein, genetic protein, protective proteins. Classification based on composition. Simple proteins. According to definition, they contain only amino acid, albumin, globulin, likewise. Conjugated proteins. They are combinations of protein with a non protein part. Conjugated proteins are as follows: glycoprotein containing glycogen, lipoprotein containing lipid along with protein. Derived proteins. They are degradation products of native proteins. Progressive hydrolysis of protein results in smaller and smaller chains, like protein into peptone, into peptides, into finally amino acids. Larger protein to peptone to peptide to smaller amino acids. Classification based on the shape. Globular proteins, they are spherical or oval in shape. Fibrous proteins, the molecules are elongated or needle shaped. Classification based on nutritional value. Nutritionally rich protein, they contain all the essential amino acids in the required proportion. Poor proteins, they lack in many essential amino acids. So, classification of proteins, classification based on function, classification based on composition, classification based on shape, classification based on nutritional value. Transamination. Transamination is the process in which amino group is transferred from amino acid to keto acid and forming new amino acid. Amino acid 1 plus keto acid 2 form amino acid 2 plus keto acid 1. Example, amino group is interchanged between alanine and glutamic acid. In almost all cases, the amino group is accepted by alpha ketoglutarate acid, so that glutamic acid is formed. In almost all cases, the amino group is accepted by alpha ketoglutarate acid, so that glutamic acid is formed. Enzymes take part in the reaction known as aminotransferases. These enzymes have pyridoxal phosphate as prosthetic group. The reaction is reversible. Biological significance of transamination. First step of degradation of amino acid. In this first step of degradation of amino acid, amino group is removed and given to the alpha ketoglutarate to form glutamate. So the transamination becomes first step for the degradation of amino acid. In this first step of degradation of amino acid, amino group is removed and given to the alpha ketoglutarate to form glutamate. So transamination becomes first step for the degradation of amino acid. Synthesis of non-essential amino acid. By transamination, non-essential amino acids can be synthesized. It is done by transferring amino group to keto acids. For example, when pyruvate gets amino group by transamination, it is converted into alanine. Those amino acids which cannot be synthesized in this manner are therefore essential. They should be taken into food. Interconversion of amino acid. If amino acid number 1 is high and number 2 is low, the amino group from number 1 may be transferred to a keto acid to give amino acid number 2 to equalize the quantity of both amino acids. This is called equalization of the quantities of the non-essential amino acids. Clinical significance of transamination. Aspartate aminotransferase, aspartate aminotransferase and alanine aminotransferase are induced by glucocorticoid which favor the gluconeogenesis. So AST and ALT are the markers of liver injury. Aspartate aminotransferase, alanine aminotransferase are induced by glucocorticoid which favor the gluconeogenesis, which means conversion, the formation of glucose other than carbohydrate, breakdown of fats. So, the AST and ALT, the aspartate aminotransferase and aspartate alanine aminotransferase are markers of liver injury. Transdeamination. It is the process by which a free amino group is released from amino acid. After transamination, oxidative deamination occurs. Transamination takes place in the cytoplasm of all the cells of the body. Amino group is transported to liver in the form of glutamic acid. It is finally deaminated in the mitochondria of hepatocytes and release free ammonia. Oxidative deamination of glutamate. Only liver mitochondria contain glutamate dehydrogenase. Only liver mitochondria contain glutamate dehydrogenase. It deaminates glutamate to form alpha ketoglutarate and ammonia. It deaminates glutamate to form alpha ketoglutarate and ammonia. So all amino acids are the first transaminated to glutamate, which is then deaminated. We call transdeamination. Glutamate dehydrogenase reaction is a final reaction which removes the amino group. The hydrolysis of glutamine also produces ammonia, NH3. This occurs mainly in the kidney. In kidney, NH4 plus production, ammonium ions production occurs, which is helpful for acid base regulation. Minor pathways of deamination. Ammonia may be formed in the body through minor reactions like oxidation of monoamines by monoamine oxidase. Ammonia may be formed in the body through minor reactions like oxidation of monoamines by monoamine oxidase. Non-oxidative deaminations. Dehydratases act on amino acid to remove ammonia from the following amino acid, that is serine and theronine. Non-oxidative deaminations. Dehydratases act on amino acid to remove ammonia from the following amino acids, serine and theronine. Desulfhydrase removes ammonia group from cysteine. Desulfhydrase remove ammonia group from cysteine. Urea cycle. The cycle is known as Krebs Henselite urea cycle. The cycle is known as Krebs Henselite urea cycle. As ornithine is the first member of the reaction, it is also called as ornithine cycle. Ornithine is the first member of the reaction. It is also called ornithine cycle. The two nitrogen atoms of urea are derived from two different sources, one from ammonia and other directly from aspartic acid. Formation of carbonyl phosphate, one molecule of ammonia condenses with carbon dioxide in the presence of two molecules of ATP to form carbonyl phosphate. One molecule of ammonia condenses with carbon dioxide in the presence of two molecules of ATP to form carbonyl phosphate. Further cycle is described below. So here it is the cycle. So one molecule of ammonia condenses with carbon dioxide in the presence of two molecules of ATP to form carbonyl phosphate NH2COOPO3 in the presence of an enzyme called carbonyl phosphate synthetase. Number one, here it is. Number one, carbonyl phosphate synthesis. So one molecule of ammonia condenses with carbon dioxide in the presence of two molecules of ATP and that convert into two molecules of ADP plus phosphate ion utilization of energy in the presence of carbonyl phosphate synthetase. What is formed? Carbonyl phosphate. So then carbonyl phosphate form citrulline in the presence of ornithine transcarbamylase. Carbonyl phosphate turns into citrulline in the presence of ornithine transcarbamylase. So then citrulline. Citrulline converted into arginosuccinate. Citrulline converted into arginosuccinate. This arginosuccinate conversion, citrulline conversion into arginosuccinate in the presence of enzyme arginosuccinate synthetase. Arginosuccinate synthetase. Arginosuccinate synthetase in the presence of arginosuccinate lyase, it convert into arginine. So arginine is going to form urea. Arginine is going to form urea by an enzyme called arginase. Along with the urea, there is another component is formed ornithine. So this ornithine along with the citrulline, ornithine plus citrulline is going to form arginosuccinate. Arginosuccinate will convert into arginine. Arginine in the presence of arginase enzyme form urea and ornithine. So remaining ornithine combined with citrulline. Okay, remaining ornithine combined with citrulline. So then next that arginosuccinate. So urea cycle shown on the left side, enzymes 1 to 5 are the members of urea cycle. Cycle on the right side is a part of citric acid cycle. This is the citric acid cycle. Then the regeneration of aspartate needs this part of citric acid cycle. Regeneration of aspartate needs this part of citric acid cycle. The two cycles together are called urea bicycle. Reactions shown in pink area are taking place inside the mitochondria, this area. Inside the mitochondria, oxalic acetic acid conversion into malate. Reaction shown in pink area. Okay, other reactions are in cytoplasm. So here, let us see here. What are the reactions here? Arginosuccinate may convert into fumaric acid. Fumaric acid form malate. Malate can form oxaloacetic acid. Then that form aspartic acid. Aspartic acid. Then aspartic acid participate in the formation of arginosuccinate. Arginosuccinate. Compartmentalization. First two enzymes are in the mitochondrial matrix, others are in cytosol. Energetics of urea cycle. The overall reaction may be summarized as ammonia plus carbon dioxide form aspartate that leads to the formation of urea and fumarate. Urea and fumarate is formed. During these reactions, two ATPs are used in the first reaction. Another ATP is converted into AMP and PPI in the double phosphate ions in the third step, which is equivalent to 2 ATPs. The urea cycle uses 4 phosphate bonds. However, fumarate formed on the fourth step may be converted into malate. Fumarate formed on the fourth step converted into malate. Malate, when oxidized to form oxaloacetate, produce 1 NADH equivalent to 2.5 ATP. So net energy expenditure is only 1.5 energy phosphates. The urea cycle and tricarboxylic acid TCA cycle are interlinked, and so it is called urea bicycle. Regulation of the urea cycle, force regulation, it depends on protein content of the diet. During starvation, the activity of urea cycle enzymes is elevated to meet the increased rate of protein metabolism. Fine regulation, the major regulatory step is by CPS1. Disorders of urea cycle. Deficiency of any of the urea cycle enzyme would result in hyperammonemia. Further results in encephalopathy and respiratory alkalosis. Clinical symptoms include vomiting, irritability, and severe mental retardation. So then 1 carbon metabolism.